Hello all and welcome to the Patient-Centric Sampling Interest Group webinar for February. This is our second event of the year and the second one of 11 planned events for this year. So we've got an exciting agenda ahead, bringing you various different usages and aspects of patient-centric sampling. Our speaker today is Ciprian, Mihai, Cirtiu, and I apologize for my pronunciation, Ciprian. He's going to talk to us today about how can a drop of blood be used for elemental analysis. But before Ciprian starts, I'd just like to quickly thank our sponsors, without whom we couldn't bring these webinars to you. And obviously, the evidence base supports us in being able to do this as well. So thank you to all of these sponsors for making it possible. Just a reminder, my name's Neil Spooner. I'm the chair and co-founder of the Patient-Centric Sampling Interest Group. Ciprian will be speaking to us for about 40 minutes. There is then going to be plenty of time for questions and answers. Please use the chat function to type in your questions and I can read them out at the end of the event. Please note that the webinar is being recorded and it will be available on both the evidence base and the Patient-Centric Sampling Interest Group websites after the event for free. So if any of your colleagues have missed it, it will be available for them to watch. If you need to re-watch it for any reason, please do so through the recording. So with no further ado, I'll go away and hand over to Ciprian.
Great. Thank you, Neil. Thank you for the introduction. Hello, everyone. Let me start by sharing the screen. I hope it works. Can you see my screen?
It's perfect, Ciprian. Yeah, it works well.
Great. So let's start. Thanks again, Neil, for giving me the opportunity to present today. It's part of a research project, but I also give you the opportunity to discover more what we do here at the Centre de toxicologie du Québec. So once again, my name is Ciprian Mihai Cirtiu. Neil, you got a very good pronunciation, so don't worry. I'm a PhD chemist and head of a trace metal team at the Centre de toxicologie, which is part of the INSPQ, Institut National de Santé Publique du Québec. I also hold a position as an adjunct professor at Université Laval and the Université du Québec at Trois-Rivières. So it's my pleasure today to entertain you on how a drop of blood can be used for elemental analysis. So first of all, I have to make this statement as I work for a governmental body. So I have no financial interest and I receive no royalties or research grants from a commercial company. Here is the outline of my talk. So I'll go with a quick presentation of the Toxicology Center. Then I will bring you into the introduction, so everything related to the work presented here. Then I will present the goals of the study. I will go into details with the method development and validation, which make use of VAMS devices. And I will show you also a few applications, especially related to biomonitoring studies. And I will finish by discussing some challenges and future perspectives. And of course, I will wrap up with a conclusion. So here we are. CTQ, which stands for Centre de toxicologie du Québec, is one of the two laboratories of INSPQ, which is Institut National de Santé Publique de Québec. We are located in Québec City, which is about 3 hours drive from Montreal. We're a public laboratory. We provide analytical services, especially analytical toxicology expertise, mainly to the health network, so the hospitals, clinics, physicians, and so on, public health agencies, poison center as well. researchers, collaborators, and so on. We develop and validate specialized analytical methods, mainly to allow assessment of exposure to various substances. So basically, we do this kind of analysis which are not done in the hospitals or other clinics. We also have the mandate to provide toxicological screening for the poison center for illicit drugs, pharmaceuticals, and metals as well, or toxic elements. One of our department also organize external quality assessment schemes, mainly for toxic and essential elements in different matrices like whole blood, urine, serum or plasma, hair, and so on. And all these activities are actually regulated by guidelines ISO 17025 and 17043. So we got accreditation for these two standards. Cirtiu, the laboratory is equipped with an impressive instrumental park, which is mainly based on mass spectrometry technologies, either for organic and inorganic, so either HPLC, LC, GC, ICP-MS, triple quad, and so on. Even tandem techniques like GC ICP-MS or HPLC ICP-MS, and so on. Let's see a bit about metal analysis. As I said, it's part of what the activities we're performing here. So when does the analysis of metals is requested actually? First of all, is in a context of acute poisoning for the poison center. So it's very important to see if someone is exposed to metals. As we know, metals could be very, very toxic. Unfortunately, this is the case most of the time for children, babies, but not only, adults as well. But we also use this assay, this test for health assessment, either related to disease diagnosis or not, especially when physicians want to assess the essential elements, or in some diagnosis like Wilson disease and other kinds. So it's very important to measure the metals in different let's say body fluids. Physicians also request metal analysis for medical treatments. So in the case when they need to monitor patients, either for medication, we know lithium is one of those elements, but also gold and other. But some metals are, for example, related to prosthesis usage. So especially specialists might request analysis for cobalt, chromium, titanium, vanadium, and so on. So all these elements related to prosthesis might be requested by the physicians. Also, we perform analysis for metals in a workplace screening scenario. So we know workers could be highly exposed to different contaminants, including metals. So for some companies, we are doing a kind of regular checkup to make sure that those workers are not too exposed, and so they don't need to get retire from the field. But one of the biggest part of the analysis we do here at the Toxicology Center, actually, it's related to biomonitoring studies. The most important in terms of number of samples we're analyzing, 1,000 per year, is the CHMS study, which is the Canadian Health Measure Survey, which is now at the 7th cycle. It's the equivalent of enhanced studies in United States, but we also did or do other biomonitoring studies like MIREC, FNBI, Esteban, and so on. And finally, we do perform a meta-analysis in investigation done by public health authorities. Of course, when analyzing metals in blood, the classical blood sampling method is the venipuncture or phlebotomy. It comes with some limitation of drawbacks, especially related to the volume needed. So sometimes it requires 5 to 10 milliliters of blood. This becomes very challenging for newborns, fearful children, or adults. For other kind of patients like extensive burns, obesity, those subjected to thrombosis, elders, and so on. But also we have to mention the high transport and storage cost, because those samples must be refrigerated or even kept frozen until the analysis. So this requires, let's say, dedicated instrumentation like fridge and freezers. But it's also an invasive method. We all know it's not easy to to get a venipuncture on ourselves. So it's painful, it's stressful, and also it requires qualified staff. As an alternative method to venipuncture, we do have micro collection tubes, capillary tubes, as well as filter paper, dry blood spot, also known as a dry blood spot or DBS. All these alternatives are suffering of different inconvenience like clots or anticoagulants. Clots because of the use of the lack of anticoagulants in this kind of devices. But also contamination, which is the main, I would say, the main drawback of, especially for filter paper. For the same technology, it's well known that hematocrit effect may play a very important role, even if related to the distribution of analytes. So this is not very useful. And another, let's say, inconvenient is the availability, because these are not available in all hospitals or clinics, so it's not easy to use them. As an alternative, recently there has been VAMS devices developed by Neoterix, which is now part of Trajan Scientific and Medical. And this new technology is based on the DBS technology, on dry blood spot technology. How does it work? So it's a polymer tip fixed on the handle, as you can see on the right image. It allows to collect precise volumes, either 10, 20, or 30 microliters, by finger prick or heel prick, especially for babies, for newborns. And it has several advantages that I will mention quickly. As you can see, this sampling approach can be done by itself. It uses this kind of devices that I will show. It comes even in kits or sampling kits. So let's see what are the main advantages, even if this is not, let's say, an exhaustive list of the advantages. I would like to emphasize the most important. So first of all, it's quite easy to collect samples, so blood sample. It doesn't require qualified medical staff. And obviously, this allows remote sampling, either at home or on the field. Let's say you have a project running in a remote community, so you might use it. Obviously, it's minimally invasive, and by this way, it's more socially accepted. So it allows to collect precise and reproducible quantities of biological fluids. We use here for whole blood, but it can be used for saliva, for urine as well, water, and so on. So other kind of liquids. And it comes in collection kit. with 2, 3, or 4 Mitra devices. A full collection kit is as shown here on the right side, the lower picture. So it comes with everything needed for sampling. So you have clamshells, you have bandage gauze, and everything you need to collect yourself the sample. One of the other advantages is the full use of the tip. So the small volume you collect, either 10, 20, or 30 microliters, can be used in one shot. So by this way, there is no classical effects related to hematocrit. So this is a huge advantage. Also, it allows storage and transport at room temperature and higher temperature, as I will show shortly. So this is available for most of the analytes, either organic or inorganic. And since the handle is it's similar to a pipette tip, so it allows to possible automate any analytical process in the laboratory, which is a huge advantage as well. And lastly, these devices are registered with health agencies here in Canada and other countries as well, so can be used even for a clinical assay. So as I said, it's easy to sample with Mitra devices. Let me run a quick video, a short video here. It's only about 3 seconds.
One drop of your blood can make a world of difference. A smarter sampling event, and that's been the.
And that's it, as easy as you can see here in the video. So a finger prick, and then you collect the small volume of the sample on the Mitra VAMS. You let it dry, and everything is done. Then you send to the laboratory for the analysis.
Can make a world of difference.
How this started, how the technology, the dry blood spot technology started. So we have to go back in the 80s, where the first papers came out reporting on the use of paper filter, dry blood spot for analysis of either organic or inorganic compounds. And it has been shown that this technology, actually progressed very well, and we can notice an exponential growth of number of papers using this technology. But Mitra VAMS has started less than 10 years ago, and it's actually showing the same trend, especially for organic substances. So this kind of substances have been more and more studied. So there are different papers on drugs analysis, perfluorinated compounds, PFAS, and other micro and macromolecules, molecules like lipids, amino acids, and so on. So there is a lot of literature on organic substances, but less on inorganic substances. Actually, the first paper using Mitra VAMS devices has been published in 2016 by Bolea Fernandez, who developed a method for a couple of elements which are well known as prosthetic metals. However, I have to mention that the method has never been used actually for real patients. So it has just been shown the potential to develop a method for prosthetic metals actually for these patients. Then a second paper by the same group has been used for cobalt measurement in patients with prosthesis. So this method has been applied for real patients actually. And recently, 2022, a biomonitoring study has been using Mitra VAMS devices for blood measurement, so mercury measurement in whole blood. And a very recent paper, end of 2023, has appeared on the toxic and essential elements in blood, actually, which is similar to what I will be showing today. On our side, we started working with DBS technology in 2018. And the first attempt was to develop an ICP-MS method for toxic elements in blood sample on dry blood spots or on the Whatman paper, especially in a forensic context. So at that time, we were targeting some elements and the forensic context. Then 1 year later, we tried to improve the previous method and to bring it at a level where it could be used in a clinical context. But we have noticed a few limitations with the dry blood spot, especially contamination, poor reproducibility of the results, and other disadvantages. And at the time, I had a couple of VAMS devices here in laboratory, and I asked the student who was working for a summer project to test those VAMS devices to see how they work. And we noticed that it's much more positive and promising. So we did then a comparative study of dry blood spot with VAMS devices. And finally, we adopted VAMS devices for the future developments. So we started in 2021. Obviously, there was a pandemic in 2020, so it was not possible to have students working on projects. But we started in the fall 2021 for a master project. And the goal was to develop and validate a method for toxic and essential elements in whole blood, obviously collected on VAMS in a clinical context. So this is the historic of how we started working with microvolumes here at the Ciprian. The goals of the study was first to explore the capability of VAMS as an alternative for blood sampling. So obviously, in our mind, we always think that it would be useful for routine and emergency analysis, as well as biomonitoring studies, workplace screening, and even neonatal screening if it works well. And the second objective was to develop and validate an analytical method for some toxic and essential elements. Here is the full panel. Among them are aluminum, arsenic, cadmium, but also essential elements like copper, selenium, and zinc. Let me bring you to the method development and validation. So we thought that the approach has to be simple and easy, right? So for that reason, we just imagined that the workflow would be just collecting the sample, blood sampling, extract the analytes, dilute if necessary, and then analyze onto the ICP-MS. So why do we use ICP-MS or tandem mass spectrometry? Well, first of all, for those who are not familiar with inorganic mass spectrometry, either simple quad or triple quad ICP-MS allows to separate, identify and quantify the elements in the plasma. So it's a hot plasma, and it allows to burn, to destroy the sample, let's say, to convert into ions, which are then quantified. This technology allows to get very good sensitivity, so very low detection limits for some elements, even PPT. For most of them, sub ppb. And there are other advantages like the multi-elemental capability. The first measurements, because you can, for a method, let's say with 40 elements, you can run a sample in less than 4 minutes, so which is quite fast, but also its versatility. But the MS-MS has been chosen because of its great capacity to eliminate interferences. And this is the main drawback of ICP-MS in general. So if we compare single quadrupole with triple quadrupole technology, so in the first one, the single quad, you'll have all the ions entering the cell, and even if you have the reaction cell, you might have your analyte reacting or the interferences, but you cannot get rid of all the interferences. So ultimately, the detector will measure the analyte of interest, but also some interferences. When using a triple quad technology, well, you have an additional quadrupole, so Q1, which might be used as a mass filter. So it allows to reject either of mass ions or interferences. So then you can use the cell to react the analyte or the interferences, and in such a way you can get rid of the interferences, or to make an adduct for the analyte of interest and to shift the mass, and then detect only the analyte of interest. So this is the huge advantage of using the tandem mass spectrometry. For the method development, so we went through first instrumental parameters optimization. For that, so we played around the gases we can use. Either we tested hydrogen, helium, ammonia, oxygen, and so on. We played around the dwell time, the internal standards as well. So we tried to have the best match internal standards and analyte. We also checked on the VAMS contamination, so the pre-treatment of the VAMS. And yes, we figure out that for some elements there is a kind of contamination. We tried 2 different approaches, chelating agents or acid to get rid of those contaminants. For some of them it works very well, for others less. We also check the effect of washing, because yes, of course, we want to reduce the contamination, but we don't want to alter the capacity of the VAMS devices, the absorption capacity. So we want to make sure that they are intact after washing, after lowering the levels of contamination. So these are the targeted elements. There is a comparison table of results, so quantification before and after washing. For most of them, we can see that it works very, very well. Arsenic, cadmium, cobalt, copper, mercury, manganese, lead, selenium, uranium, and zinc has a positive impact once we wash the VAMS. I also reported the reference values in blood for these elements, so just to show that there is no impact even if there is still a very low amount of contamination after washing. But unfortunately, for some elements like aluminum, chromium, and molybdenum, it was just not possible to lower the contamination level to such a point where there is no impact on the quantification. So we abandoned these elements, and we continue with a panel of 10 elements, toxic and essential elements. Then we also check the sample extraction process. We tested for extraction with different agents, but also digestion, and we figure out that digestion is the best way to recover most of the analytes with a good recovery, actually. We also test for a different calibration approach, and we figure out that matrix match calibration is the best approach, so we choose this one. And finally, we test for stability at room temperature as well as 37 degrees. Why testing at 37 degrees? Well, here in Canada, it's probably not the case, but in other countries, hot countries, like in Africa, for example, when you need to run a project in the field and you cannot store those devices, let's say, at room temperature or around 20 or even lower, 4 degrees, when you have to consider this aspect, the stability at the higher temperature. So for that reason, we tested in parallel with room temperature 37 degrees. And finally, the last step in method development was the instrumental parameters optimization. And here are the final option, actually, the gases we choose for each element, as well as the transition and the internal standard, optimized internal standards for each of the elements. Then we move to the validation of the method. As I said, our laboratory is accredited for ISO 17025, so we follow these guidelines for validation. So we did tests for LOD, LOQ, linearity range, and matrix effects as well, robustness, repeatability, and stability over time at different temperatures. Let me show you here the method parameters. So let's start with the limit of detection and quantification for the validated elements. Most of them show very good or very low detection limits. Obviously, when we compare with the standard method where we only dilute the sample, let's say, and we shoot onto the ICP-MS, we might expect lower detection limits for the dilute and shoot approach instead of digest and dilution, as it's the case here with the VAMS. So for most of them, we have comparable detection limits or slightly higher than conventional methods, but still very sufficient for quantifying and providing reliable results for those elements in whole blood. Then we determine the linearity range and the matrix effects. So we just wanted to make sure that everything works fine when you change from one type of blood to another one. So there is no change in the quantification or impact on the level quantification. So here are the parameters. It works very well, actually. All these data are already published in the paper cited below. So don't worry if I don't go into details. You can consult that paper, and you'll have more details and interpretation of the results. We also assess the repeatability for these elements at two different levels, with a low QC material as well as with a high QC. It works very well for most of them. And finally, the robustness test for the validated parameters. So this test allows to assess the impact of slight variation in different experimental parameters, like digestion time, cleanliness of the VAMS devices, dilution factor, or a brand of nitric acid. For most of them, it works very well. The acceptable range is 10%. We can notice that in some cases, like for cadmium, the dilution factor might have an important impact. So this might affect the results, but again, it's just a deviation from the normal standard protocol we might apply in the laboratory. So for lead as well, as you can see, the cleanness might impact the results as well as the dilution factor. So there are some factors that once we develop and validate the method with these parameters, so we have not we should not actually deviate too much, because the results ultimately will be impacted. Then the last parameters we assessed is the stability at room temperature. So we did for 1 month, 2 months, 3 months, 6, and finally 1 year. Most of them are performing very well, are very stable over time, even after 1 year. For some of them, you can notice some change, some variation, like for arsenic, for example, and cobalt. And this is actually more related to the analytical aspects and not stability as itself, actually. Because arsenic, we noticed that over time, we might get, let's say, a kind of contamination over time. And by applying a blank subtraction, because all tests were run with a blank, actually, and that blank has been subtracted. So by applying a higher blank, so we subtract the results are obviously impacted, and that's why we get let's say a loss of arsenic after 1 year. But roughly after 6 months, we are still quantifying with a very good results. So we're not worried about. For cobalt, it's about the same thing. But as I said, most of them are very, very stable over time. The second graph is showing the stability at 37 degrees. Again, a lot of variability with arsenic, which is not an easy one. But otherwise, for all the other elements, it's very stable after 4 months. Mercury is showing a bit of variability as well. The same reason we might expect some variation, either due to contamination over time increase. In some papers, we have seen a kind of increase over time due to contamination, storage contamination. It is not yet clear. We did some tests on our side, but didn't conclude that might be some contamination. We might just simply use, because we know mercury is quite volatile, and at 37 degrees for 4 months could be very dangerous. So, but most of them are very stable. So we got good results, variable results for cobalt and mercury, as well as arsenic in some conditions. Manganese is not shown here, because at the time when we performed this test, the lot of meter devices we have been using were highly contaminated in manganese. So it was not possible to monitor that element, actually. But we got then good, let's say, good meter devices without manganese contamination. And it allows to validate actually the method, fully validate the method for manganese. But unfortunately, the stability test it's not yet completed. So roughly, most of them are very stable at room temperature, as well as at 37 degrees. Then we compared the VAMS versus venipuncture, so the collection approach, but also the analytical methods. So we compared our new methods based on VAMS collection with a routine analysis method used here in our laboratory based on a dilution and injection approach. So we compared different things at the same time, and we did this with a cohort of 50 participants, unexposed people, so healthy and unexposed people. And the comparison was not that easy, actually, because of the low levels we have been measuring. So we tried to separate the results based on in 4 different groups. So either if the results are similar, if there is a difference less than the detection limits or the quantification, as well as inaccurate results. But this is not a straight comparison. It's since we are using very, very low levels, so it's really tough to compare. and most of them, like for example uranium, all the results were under the detection limit, so it's just not possible to compare actually. But for that comparison, so we pushed the study forward, and we used the Spearman rank correlation, and for 6 of those elements where detection rate was higher than 50%, so we got a significant monotonic correlation for arsenic, copper, mercury, selenium, lead, and zinc, and this is very promising actually. Then we did apply this new method and this new approach of sampling to real, let's say, the real application in a biomonitoring study. The first one is I want to share with you is a UNICEF tender. In 2022, actually, January 2022, UNICEF published a tender related to public health issues in Georgia, in East Europe, actually, related to lead exposure. The objective of that tender was to the establishment of a national system for environmental health surveillance in Georgia, a GLES program. And why that? Because children were highly exposed to lead. There was a previous study in 2018, a mixed study, a multiple indicator cluster survey, who shown that those children are highly exposed to lead. Obviously, lead is a neurotoxic agent, especially for young children and women of childbearing ages. But also there is no safe threshold, either for children or adults. And so we jumped in with collaborators in Norway. So we did join an international consortium with the Norwegian Institute of Air Research, as well as Arctic University of Tromsø in Norway. And our contribution actually was mainly based on the analytical section. So everything related to blood analysis and blood isotope ratio as well. So for that, we started developing methods. But what this mixed study shown is that 62% of children has a blood lead level higher than 3.5 microgram per deciliter, and almost 20% higher than 10 microgram per deciliter, which is quite high. Ajara, which is one of the part of the area of Georgia, has the highest contamination level, followed by Guria, Samegrelo, Zemozaventi, and so on. Other regions have a high level of blood lead level actually when compared to other countries. There was no difference between rural and urban areas, excepting for Ajara. And there are other details that can be shown in this study. But the main conclusion is that the source of exposure is not yet identified. So this was let's say the goal of the new study is to identify the source and to put in place a program for monitoring those children. Mixed study also performed some isotope ratio measurements for lead, so LIR measurements. As we know, lead is stable, has some isotope very stable in the nature, but also radiogenic and cosmogenic. So we can use it to make to establish the profile of lead and to compare to different references. As for example, the study has shown that all the ratio determined in those blood samples seems to indicate a single and unknown source of exposure for lead. So we don't know if it's the food, the environment, water, soil, and so on. So for that reason, so we needed to develop different methods. First, a simple and transferable method to another laboratory, obviously on the same instrument or a similar one. And for that reason, so we started working specifically on lead. We did develop and validate the method, and then we implemented in their laboratory, and we provided training to the team. So that work has been published in 2023, and the method is now running in the laboratory. They have sampled more than almost 2,000 children, ages 5 to 7 years, and it works very well actually. Only very positive feedback from the field actually. So there is no problem with sampling, and this is very promising and encouraging for other studies. Talking about other studies, actually there is a cross-Canada research study that has started here in Canada last year, and it's still ongoing. And for the next steps, the next phase actually, they plan to use the VAMS devices for sample for blood collection, and to analyze for cadmium, mercury, manganese, lead, and selenium in our laboratory with the full method we just developed and validated. And this is a study about the nutrition, health, and environment of the First Nation children and youth here in Canada. Okay, let's talk a bit about the challenges and future perspective. Among the challenges I want to share with you today, of course, the first one is let's say a commercial one, because there is only a single manufacturer for these devices. So if there is a contamination, for example, for the raw material, the plastic could be subject to metal contamination. So this becomes a limitation actually. Also the cost. When we compare VAMS devices to a regular sampling tube, obviously there is a difference in the cost. Let's say 10 times most costly for the VAMS than regular tubes. However, we have to consider let's say the full workflow, the full process when calculating or assessing the cost. So and comparing with the traditional way. So we have to consider collection, storing, transport, handling, waste measurement as well. So for that reason, it's not easy to compare or to conclude that VAMS devices are just more expensive than regular collection tubes. So a full exercise has to be done and consider the health economics actually related to the workflow. The sampling limitation is actually challenging. This approach might apply to most of the patient, but in some cases it becomes limited. For example, when patient suffers of dehydration, poor blood circulation, or shock condition, for example. Another limitation or a challenge for now is the use of the sample units, the total use of the sample units. So if you collect only 1 VAMS, so you cannot repeat the test. So for that reason, you have to collect 2 or more Mitra sample devices. Fortunately, this comes in a kit, as I said, 2, 3, or 4 units. So feel free to collect more than 1 unit to make sure you get a chance in case you suspect contamination or the test just didn't perform well. So you will be able to repeat the test if you have a second unit. Finally, it's worth mentioning that the quality of the results will strongly depend on the quality of the sampling. And this is true for all type of sampling, obviously, but especially here with VAMS devices. So you have to make sure that you do a correct sampling. You have to follow the instruction provided by the company. It comes with the kits, so all the kits have this sampling procedure included. You have to avoid oversampling or undersampling, because this will have a huge impact on the results, obviously by overestimating or estimating or underestimating the results. As a future perspective, I would like to mention actually our main goal, which is to implement this technology in the health network. So we're working hard on these aspects. We want to bring them as an alternative in the hospitals for blood collection. Unfortunately, it's still the case. It's very difficult to collect samples for babies, newborns, children, and other categories of patients. So for that reason, we need an alternative, and we work hard on that aspect. On our side, and I suppose it's the case for other laboratories, we might need to transfer conventional methods to microvolumes. Historically, all our methods here at the Cirtiu have been based on conventional volumes, so thousands of microliters or milliliters of samples actually. But now it's possible to go to microvolumes. So this is a future perspective on our side, actually, to just to move from conventional volume to microvolumes. And we all know that there are other emerging substances which are showing high interest, like PFAS and others. So we work on that aspect, not only on as I already mentioned, developing and validating a method for isotope ratio of lead, but also we are targeting PFAS in whole blood actually. And I really think that with the new development, the technological development, will have new versions and more choices on the market. So this will probably allow to collect other kind of samples, like serum or plasma, for example, which is not the case with the actual technology. And I'm very confident that the continuous improvement of the instrument sensitivity will allow us to actually to have more developments on microsampling, and so this will just benefit, actually, the microsampling development will benefit from the improvements in instrument performances. So this will really help to have new developments for microvolumes. Let me wrap up with a few conclusions. So I really hope that I demonstrated that VAMS could become an alternative for blood sampling for different reasons and with different advantages. So it allows to collect low volumes, precise and reproducible as well, which is not the case with dry blood spot paper, for example. There is no effects related to hematocrit, as it's the case for DBS. It's an approach which is very oriented towards the patient. So it's really a patient-centered approach, and it really applies to difficult cases. It's less expensive when considering the overall cost of the workflow. So consider all the aspects, pre-analytical, analytical, and post-analytical aspects, and not only the mitra-VAMS cost. And it shows a good stability at room temperature, as well as 37 degrees for most of the elements. So with this in mind, obviously, mitra-VAMS devices becomes an option for blood sampling in different contexts, either in a clinical one for a regular checkup by physicians, but also in a workplace scenario where we need to follow up and to ensure a screening of workers, but also in all studies, biomonitoring, biosurveillance studies related to environment contamination. So studies like ICP-MS or other surveys across the country. And the last and not the less important, I would say, it's the idea to have a kind of combined the assay, for example, for lead and PFAS, as a result of water contamination, for example. So with mitra-VAMS, it's now possible and easy to collect blood samples, and this opens the doors and allows to create science and knowledge about the impact of different contaminants on the human health. I would like to acknowledge, first of all, the audience and PCSIG group who gives me the opportunity to share these results, this study, this research with you, as well as the Institut National de Santé Publique du Québec, which obviously supported the study from a financial point of view. Neoterix, which is our partner, the company has provided free of charge the mitra-VAMS devices, and we thank them very sincerely, because it allows to generate of this new science and make progress in the patient-centered approaches. Also, we want to thank UNICEF, who sponsored part of the study, that part related to lead analysis in Georgia, as well as Université du Québec à Trois-Rivières, and Anthony Breton, who is actually the master student who worked 2 years on this nice project. And thank you, everyone, for your patience, and I'm waiting for your questions.
Thank you, Ciprian. That was a fantastic presentation. Very, very interesting. We don't have very much time for questions, but we'll try and answer some now. But I was just wondering whether, because a lot of people are now leaving on the hour, I guess.
I'm so sorry I was longer than
That's okay. That's okay. It happens. Maybe what we could do is we'll do a couple of the questions now, but
Sure.
Maybe if we can capture the questions, and if you could provide written answers, we can post those on our website along with the video. Would that be okay?
Yeah, that's fine.
Okay. So there's quite a few of the questions around contamination of the samples, and just the whole idea of background subtraction, subtracting a blank kind of thing, wanting to know about variability between lots, within lots of that subtraction, and also wondering whether you could include a blank matrix sample, or whatever technology you choose to use for your blood collection, whether you could include a blank at the time of collection so that would pick up any environmental contamination. So just any comments about that, the within lot, between lot variability for background contamination, and whether you could normalize for background contamination at the time of collection by including a blank sampler.
Sure. Let me start with the blank actually used during the test. So all the tests were performed with blanks as well. So let's say environmental blanks So if a sample has been exposed to air, for example, either during collection or preparation for the analysis, we always had a blank, a witness blank, and we reprocess in the same way. So we have been able to assess if there is any contamination, and if necessary, that blank has been subtracted. But most of the time, it was not necessary. So we just serve as a, let's say, a controller to make sure that there is no contamination during the sampling, the treatment of the sampling and the preparation for the analysis. Now, regarding the different lots of VAMS, we notice that most of them compare very well. From different lots, we get the same level. The two problematic elements have been arsenic and manganese. And at the beginning, the first lots we received were highly contaminated in arsenic and manganese. But then the subsequent lots of these devices were actually free of contamination, and washing or unwashing has no impact actually on that. Reproducibility, it's much better than what we noticed with dry dot spots actually in the past with Whatman paper, and it's lower than 3%. So we are very, very satisfied and confident with this this level of reproducibility.
Okay, thank you, Ciprian. We'll do one more question and then we'll finish because we're starting to lose our attendees now. Just something from Ross. What measures are you making to correct for incomplete or excess volumetric collection? So you mentioned that towards the end. Do you have anything that can normalize for that or easily spot it as an issue?
We do not normalize actually. So we analyze the full mitra VAMS. So we hope that the volume is as indicated, because there is a certificate coming. So the volume is certified. So we do apply that certified volume in our calculation. So obviously, results will be subjected to uncertainty related to the sampling. But if the sampling is correctly done, there is no need to correct for other effects actually.
Okay, we're going to call it a day there, Ciprian. I just want to share my screen so that just to wrap things up. So thank you for your presentation. On the 14th of March, we have our next in this year's presentations from Andrew Patterson, who's going to be talking to about PFAS, so related to today's talk, really, advancing the collection and analysis of PFAS. So if you're interested in that, please take a look on either the evidence base or the patient-centric sampling interest group website, and you'll be able to register there. Today's webinar will be available for free quite soon once we've uploaded the video. If you're interested in joining the patient-centric sampling interest group, please just take a look at our website or a LinkedIn group and you can learn more about us. Just finally want to say thank you to the evidence base for helping to host this webinar and to all our sponsors for making it possible. And finally, I'd like to say thank you again to Ciprian for a great presentation today. And thank you to everyone for attending, and we'll try and address your questions through our website in the next few weeks. Thanks all, and goodbye.
Thank you again. Thank you. Bye-bye.